Antigen processing and presentation is the process of digestion of antigens into smaller peptide fragments by an antigen-presenting cell, APCs, that are then displayed on the surface of the cells via antigen-presenting molecules like MHC class 1 and 2 for recognition by lymphocytes. Antigen processing and presentation can occur via 3 different pathways. The first pathway is the endogenous pathway or classical MHC class 1 presentation. In this pathway, the antigen-presenting cells degrade the protein antigen into short peptides using a cytosolic proteolytic system called the proteasome. The immunoproteasome, induced by exposure to interferon G or TNF-A, is involved in the immune system. The peptides are then trimmed by aminopeptidases in the ER lumen and transported to the ER lumen by the transporter associated with antigen processing, TAP. Tapesin and the calnexin-calreticulin system mediate the trimming and loading of peptides on MHC class 1 molecules. Depending on the affinity of the loaded peptides, MHC class Y molecules will either be transported to the cell membrane or recycled by UDP glucose, glycoprotein transferase 1. High-affinity MHC class 1 complexes are transported through the Golgi apparatus to the cell membrane to elicit antigen-specific CD8 plus T-cell responses. The second pathway is the exogenous pathway or classical MHC class 2 presentation. In this pathway, antigen-presenting cells internalize the antigen through phagocytosis, where the material first binds to specific surface receptors. The degradation of proteins into peptides occurs within the compartments of the cell by the endocytic processing pathway. The internalized antigen progresses through different acidic compartments, encountering hydrolytic enzymes and low pH in each compartment. The final protein degradation and peptide loading take place in a unique endosome called the MHC class 2 containing compartment, MIIC. To prevent the interaction of antigenic peptides with class 1 molecules, the APCs have a distinct mechanism. When class 2 MHC molecules are synthesized, the class 2 AB chains associate with a protein called the invariant chain. This protein interacts with the class 2 peptide binding groove, preventing the binding of endogenously derived peptides. The invariant chain is degraded until it forms a short fragment called CLIP, class 2 associated invariant chain peptide. Later, a class 2 MHC molecule catalyzes the exchange of CLIP with antigenic peptide.